This is a video lecture on Chapter 18, the immune system. There are 5 nice sections to this chapter which logically lay out the components of the immune system. People logically assume that the majority of the immune system is contained within the blood. That, as we'll see shortly, is not entirely true. The blood does contain components of the immune system, represented as 2 factors. The first are the cells. The leukocytes, the white blood cells, they are components of the immune system. But what you don't see in this figure are the proteins and other factors that allow the immune system to communicate, not only with itself, but to detect and communicate those differences with the rest of the body. Figure 18.1a shows us how by staining using different DNA stains, we can identify through the morphology of the nucleus and other granules in the cytoplasm the nature of some of these immune cells. So this is a multi-lobed nucleus, and this belongs to something known as a neutrophil. Over here we have another cell with a different profile. This will be known as a monocyte, a precursor cell that can give rise to macrophages and dendritic cells and some other types of cells of the immune system. The following 2 slides, contained in Table 18.1, are these cells, the most prominent cells of the immune system, the site at which those cells are produced, and the functions of those cells. Should the student learn each one of these? I believe they should. It's nice to know what individual cells of the immune system are and what they do. So take your time and pick 2 or 3 that you're familiar with, and then go on to learn some of the others. The color coding may help you. Table 18.2 does the same thing, but this time using chemicals known as cytokines. Cytokines are chemicals that affect the behavior of other cells. Let's turn our attention to one of the 2 major compartments of the immune system, the vertebrate immune system. So not just humans, but all vertebrates have this subdivision. The characteristic role of the innate immune system is to recognize general molecular patterns and properties of pathogens, anything that's not normally present in a higher vertebrate. In multiple textbooks on immunology, there is a subdivision of the innate immune system into the first line of defense and the second line of defense. In our textbook, we consider that as a combined category of first line defense. That includes the defenses embedded at body surfaces. Don't just think of skin, think of any surface inside or outside that's lined with epithelial cells, such as your digestive system, your urinary system, your reproductive systems, your respiratory system, etc. The process of inflammation is also regarded as a major component of the innate immune system. The utilization of chemicals known as interferons is critical to communication about the state of infection and regulation of the innate immune system. The 4th category are the receptors on some of the immune cells which are able to recognize foreign substances. One class of those very important receptors are known as toll-like receptors, or TLRs. The first line of defense would be the barriers, the physical barriers and chemical barriers that our body possesses. Skin comes to obvious mind, but remember, mucous surfaces themselves are also barriers because the outside world extends through your various openings into your body. In this list, we would have to include the natural flora and fauna that populate our skin in various locations, as well as the secretions that are produced, including sweat, which contains very important fatty acids that are antimicrobial. Tears and saliva are included in that list of chemicals. For instance, the enzyme lysozyme appears to be very important in the majority of secretions in the body. Other substances would include peptides of various classes found in these fluids. With respect to inflammation, there are 4 factors that one has to consider. These would be redness, due to the increased flow of blood in that region, swelling, caused by the passage of plasma out of blood vessels into the tissue, heat, again because of the delivery of extra fluid from deep within the body, and pain, to alert the body of the consequences of that infection or wound. Local inflammatory responses are pretty complex in their entirety. They normally begin by the detection of pathogen-associated patterns by cells which are parked or residing in tissue close by to the site of the wound. These cells then release chemicals, as we'll see on the next slide, that initiate the permeability and therefore the extravasation of white blood cells from the nearest blood vessels. And as those white blood cells and the plasma associated with them enters the local tissue, delivery of extra white blood cells and their factors takes place, therefore tackling or at least containing the infection. Upon clearance of the infection, the situation then settles down and returns back to the normal homeostatic state. Communication is key, so without these mediators of communication, the voices, in quotation marks, of the immune cells would never be heard. For instance, kinins are released by enzymatic actions on plasma proteins, and their intervention entails the dilution of blood vessels, therefore increasing the permeability, leading to inflammation. Histamine release by mast cells and some types of injured cells leads to increased vascular permeability, resulting in the recruitment of more immune cells. Please make yourself familiar with the rest of this table. Figure 18.3 presents a nice figure showing the relative size of pathogens in red and the immune cells, in this case a macrophage, in brown. The bacteria are being contacted by processes of the macrophage, leading to phagocytosis. Once internalized, the vacuole with the bacteria, known as a phagosome, will then fuse eventually with a lysosome to form something known as a phagolysosome, and that's where the death of the pathogen is initiated through the chemical action of substances contained within a lysosome. The digested products will be used by the cell or excreted into the environment. The process of phagocytosis by certain cells of the immune system is paramount for that particular mode of action. Figure 18.5 lists some of the functions of phagocytes involved in the innate system. These cells can perform phagocytosis, leading to intracellular killing of microbes. However, the contact can also initiate, at the same time, the secretion of chemicals by the phagocytic cells. And these chemicals are very important in leading to many outcomes, ranging from developing fever, to extracellular release of killing agents, to the control of the inflammatory response, and finally to clotting or anti-clotting pathways, leading to the delivery of other components. The other arm of the innate immune system that's very important is that of complement. Complement is a system of proteins, most of which are produced by the liver. The role of these proteins is to attach to and then lyse foreign cells, especially bacteria. The multiple modes of action of complement is not the subject of this chapter. All you need to know is what initiates complement and what the outcome of complement activation is. So there are a number of proteins, all starting with the letter C, C1 being one of those important initiators, and the other being C3. Once the complement cascade, as it's known, is initiated, it can lead to a number of different outcomes. The most common is the development on the surface of the bacteria of a multi-unit protein complex known as the membrane attack complex, MAC. The MAC looks like a giant donut on the surface of the bacteria, and in some cases the viruses, and that, once completed, forms a channel that allows material to cross over through the cell wall and the cell membrane of these bacteria or viruses, permitting ions and water and small molecules to migrate to either side, therefore disrupting the intracellular environment and thus killing the microbe or disabling the virus. Another form of activation of the complement pathway, an alternative, is by binding to the surface of pathogens using C3b. The C3b then can bind in an allosteric fashion with the C3b receptor on certain phagocytes like macrophages. That encourages these cells to easily phagocytose the target cells in a process known as opsonization. Figure 18.7 relays some of the known functions of complement proteins in the human body. So the first path is direct destruction by forming the MAC complex. The second mechanism is vasodilation and increasing permeability of capillaries and venules to proteins, and those proteins may be bactericidal. The chemotaxis of cells to the location where the complement proteins detected the presence of pathogen by binding. And of course, as we just spoke about on the previous slide, the enhancement of phagocytosis by coating the surface of the pathogen. Another defense mechanism present in all cells is that of the ability to release type 1 interferon upon infection by certain types of viruses. So the cell itself cannot be prevented from death, but it can certainly help its neighbors by releasing the type 1 interferon into the extracellular environment. The goal is to change the biochemistry of neighboring cells in such a way that it makes the replication of the virus, should that virus obtain its entry into those neighboring cells, a bit more difficult, and in some cases block replication. The type 1 interferon can also act in an autocrine fashion, where it can change the behavior of the virus-infected cell to delay or slow down the propagation of progeny viruses. There are many aspects of the adaptive immune system that we don't have figures for, but the keyword list at the end of the chapter is a great way to cross off those areas that are mentioned. Let's migrate our attention now to the adaptive immune system. The major difference between this and the former is that this is designed to attack specific foreign material. So based on a number of amino acids or a few polysaccharides, that's all it takes. Thus, any of these molecules that can trigger an adaptive immune response is called an antigen. So antigens is easy to remember because antigens bind with antibodies. Regardless, a typical adaptive immune system response can be divided into 3 steps: the recognition, the activation of the white blood cells, and then the attack that's launched by these activated lymphocytes. Unfortunately, the process can take up to a week to perfect and launch. These attacks are normally launched from the secondary lymphoid organs. Those include the spleen, the general lymph nodes, the tonsils, and the adenoids just close by. Other places that offer a close association between the lining of the body and the pathogens that could gain entry through those places. Those include the Peyer's patches in the intestine, the respiratory tract, the genital system, and the urinary tract. Bone marrow and the thymus gland are classified as primary lymphoid organs because that's where these cells, the B cells and the T cells, are maturing. Figure 18.9 gives a generalized view of a typical lymph node where lymph enters in one direction and then it leaves once it percolates through this organ in another direction. There are valves that are one way, just like we have in veins, and they prevent backflow of the lymph. Each lymph node is broken into sinuses, and the sinuses are simple gaps between the structural cells that make up the organization, the anatomy of the lymph node, and the populating B cells and T cells, which are resident in the lymph node. They can travel through the circulation, but only when they are mature or when they first have to arrive to populate the lymph node. B cells in particular generally don't leave once they arrive in the lymph node. T cells are wandering constantly back and forth. There is a sophisticated interplay of cells and chemicals that is a bit too aggressive for this book. B cells and T cells are all derived from precursor cells in the bone marrow, stem cells. They then differentiate depending on their geography and the signals that they receive along these lineages. T cells, premature T cells, will migrate to the thymus where they complete their maturation. And then they will go and populate the secondary lymphatic structures. B cells will do that directly from the bone marrow. And once in the secondary lymphoid structures, T cells will then differentiate into several classes, and the B cells will then lie silent until they are activated by antigen and start releasing huge amounts of antibody. This figure does a great job of summarizing the general functions of the two classes of T cell and the one class of B cell. The important thing is that the helper T cells, their role is to help activate both B cells and cytotoxic T cells. And once activated, those cells then go to work in fighting infections. Concentrate briefly on the general organization of an antibody molecule. An antibody is a protein, and these proteins are made by B cells. They may be present on the surface of a B cell and also released into the liquid part of the extracellular fluid. Antibody molecules are generally Y-shaped, such as this. They have two polypeptide chains, a heavy chain and a light chain, and the terminal part of each of these two chains is the part that's variable. So this part here, the tip, can be multifaceted in that it can be produced by different B cells in different structural arrangements, so that two different B cells are never the same. The lower half of the molecule is always constant, and that pertains to the class of antibody or immunoglobulin. Evolution has equipped our bodies with 5 classes of Ig. Ig is an abbreviation for immunoglobulin. So there's IgG, representing the majority of antibody in the plasma. There's IgE, normally found on the surface of resident cells like mast cells, basophils. Also IgA, generally exported beyond the epithelial endothelial cells. And then there's IgM, which forms aggregations. Finally, there's IgD, which has a minor role throughout the body. The cells of an individual human person are tagged with proteins that tell the immune system that these cells belong to one's self. These tags come from genes which are expressed. The genes that make these tags reside on chromosome 6 of human beings, and they are part of a cluster known as the major histocompatibility complex, MHC. And this terminology comes from just research labs that initially discovered this. Why are these important? Because these molecules are interrogated on a regular basis by T cells. And T cells will only take action if they first make a connection, a handshake, between their surface proteins and these MHC molecules. The purpose of these handshakes is to guarantee that rogue T cells don't go around activating themselves without first being informed that they are participating in legitimate reactions. There are 2 classes of MHC molecule. Class 1, which are produced and displayed on the surface of virtually all cells of the body, except red blood cells. And there's class 2, which are only found on the surface of macrophages, B cells, and dendritic cells. And these cells are antigen-presenting cells in that they can engulf through endocytosis pathogens, cut them up into pieces, and then display them on their surface. We'll see that in the next few slides. So the 2 classes are important in interacting in different ways with the adaptive immune system. The different classes of lymphocytes can then interact in various ways with these MHC molecules. So B cells are not equipped with any complementary proteins to interact with MHC, so they don't do that. Nor do natural killer cells. They are MHC independent. So which types of cells do interact with MHC? It's the T cells. So the helper T cells, the CD4 T cells, they preferentially bind to class 2 MHC molecules, which are found on these antigen-presenting cells. What the helper T cell then does, it allows or provides permission for these types of cells to continue to mature and become activated. The other class of T cells are the CD8 T cells, or the cytotoxic T cells. They bind to class 1 MHC molecules, which are present, as we said earlier, on all nucleated cells of the body. And the reason in this case is that any cell that's infected by a virus can then ask a T cell, cytotoxic T cell, to intervene and kill that cell. In this figure, we see a macrophage asking for permission to activate itself by presenting in its MHC class 2 molecule a fragment of something that it's swallowed through phagocytosis. An interesting observation is that the antigen is kept inside a phagosome until parts of it are digested and loaded onto a class 2 MHC protein and then displayed on the surface. If a helper T cell happens to recognize the antigen, then it will bind and then release chemicals that will activate the macrophage. B cells achieve the same result, but they use their cell surface antibody to bind to the antigen and then deliver that to the interior phagosome. Because the outcome of these interactions can be quite dangerous for the body, evolution has required a robust and diligent handshake between the antigen presenting cell and the activation of the helper T cells. So the first handshake is the specific binding between the class 2 MHC and the CD4 molecules on the surface of the helper T cells. The second binding involves other protein markers that are present on the surface of both cells, and once they fit, then the antigen presenting cell will have to secrete interleukin-1 and tumor necrosis factor alpha in order to guarantee that the handshake is legitimate, and that will wake up the helper T cell, which in turn will become activated and initiate other close events which are part of the immune response. Class 1 interaction, HMC class 1 interaction, takes place between cytotoxic T cells and any cell in the body that has a nucleus. And in these situations, the purpose is to rescue some of the fragments of an internalized virus and to present them through class 1 interaction, therefore asking the cytotoxic T cell to initiate the death of this infected cell. The odd cells of the immune system are natural killer cells. Year after year, their activity has been discovered or rediscovered, and their localization within the confines of the immune system tend to migrate from the innate to the adaptive and back again. Regardless, natural killer cells are cells that have been equipped to kill virus-infected cells and precancerous cells by non-specific means. Immune tolerance refers to the ability of the body to select away any T cells that will recognize self, any B cells that would also recognize self. So immune tolerance is simply a mechanism for eliminating, through deletion or inactivation, cells that would match one's own antigens. These processes are pretty complex and beyond the scope of this chapter. Figure 18:16 presents a cartoon showing in a very simplified form a summary of how bacteria would be then presented to helper T cells or to B cells, leading to some type of response. In this particular example, the activation of the B cell must have a corresponding T cell that will then give permission to the B cell to produce antibodies. And those antibodies are then released via plasma cells into the general circulation. And at the same time, some of the B cells, a very small percentage, will become memory cells that will then respond to a second infection by the same antigen faster the next time around. We mentioned the 5 classes of antibody that were made earlier in this video. Now let's have a look at how they perform their functions. So attacks coordinated by the antibodies have been observed in a number of different methods. So one is direct enhancement of phagocytosis. So in this case, the antibody binds to an antigen. In this particular example, it happens to be the cell wall of a bacterium. The binding activates the Fc portion, the constant portion of the fragment of the antibody, and then that binds to receptors on the surface of phagocytes. The phagocytes then will eat and destroy the bacterium. An alternative mechanism is to use the antibody to activate the complement system that leads to either the formation down this pathway of the membrane attack complex, leading to the rushing in of extracellular material, disrupting the functionality of the bacterium, or in an alternative pathway, the complement cascade leads to the binding of C3b to the surface of the bacteria, which permits phagocytosis of the pathogen. Additional involvement of antibodies is mentioned in number 3. In this particular case, antibody-dependent cellular cytotoxicity is mediated by the activation of natural killer cells. So once the antibody binds, a natural killer cell will come along and recognize the constant region of the antibody and then destroy the cell to which the antibody is attached. And as it says in the book, this is the one exception to the non-specificity of natural killer cells. In this case, the antibody will bind specifically to its target molecule, the antigen, and that happens to be on the surface of a cell. Therefore, that is a specific nature that the natural killer cell relies on. Finally, we have the direct neutralization of toxins produced by bacteria or virus particles. So in this case, the binding of the antibody leads to precipitates being formed, and those precipitates can be then removed from the tissue, or these precipitates can prevent the toxins from binding to something else because they're already bound to the antibody. So these are some of the mechanisms so far discovered. Figure 1819 simply shows us what we already know, that the first time you're exposed to an antigen, it takes time, a number of days, up to a week and a half, for the activation of the adaptive immune system to take place. But subsequently, if you're challenged again with the same antigen, the presence of memory cells causes a rapid and immediate response of the adaptive immune system the second time and subsequent times that antigen comes across your body. There's a great association between where an organism lives and the pathogens that are present in that environment. So this repeated exposure to pathogens results in a buildup of immunity, and that is called active immunity. Vaccines can add to the repertoire of immunity that an organism has, and vaccination relies on mimicking these natural outcomes. So in a vaccine, we have small amounts of either living or dead pathogens, normally proteins, or their toxins, and then these are delivered to the body in such a way as not to cause disease in the first instance, but to expose those epitopes to the immune system so it can generate memory cells. In contrast, passive immunity is simply using somebody else's antibodies or memory cells to supplement your own. And a baby does this very well, either as a fetus or once it's born as a baby. So we now know that the interaction between the mother's bodily fluids and that of the fetus can transfer antibodies across the placenta. Additional antibodies are delivered through breastfeeding, and additional antibodies are delivered through early life via nutrition, either by feeding or drinking. The interplay between cytotoxic T cells and helper T cells is reiterated in this figure. Helper T cells are programmed to help cytotoxic T cells become activated and then go hunting for cells that contain virus. In an earlier slide, we spoke about interleukin-1 and tumor necrosis factor. Now we're going to look at interleukin-2 and its partner interferon gamma. So activated helper T cells, once they have become activated, are then equipped to secrete both these substances. And their secretion, depending on the vicinity, will either activate macrophages, which will then go around delivering cell-killing chemicals, or they will interact directly with natural killer cells, activating them to then do the same. Table 18.6 does a good job of summarizing how your body responds to viruses and the different interplay that goes on. So you'll notice that there's not one single pathway that is used, because the body is attacked by viruses that have evolved in many different directions. Therefore, the body has to be equipped itself to fight those various intruders. It makes sense that any type of infection or injury should be battled as a whole body experience, and that's what this slide is showing us. So whenever distant relations are activated in the immune system sense, other organs are involved at the distant sites from the initial infection, we call that the acute phase response. And fever is the great example. So raising the fever has a multitude of effects on the battle between the pathogen and the body. The human body can continue to work at a higher temperature, that of fever, whereas the replication of the pathogen is normally slowed down. That gives time for the immune system to mount a sustained and specific attack against the pathogen. The liver plays a great role in an acute response because it can sequester iron and zinc, which are necessary for the replication or metabolic needs of bacteria, because they require large amounts of iron in order to continue to function and replicate. And our body doesn't need that concentration. In addition, mobilization of various substances and cells is also a part of the acute phase response. And finally, the hypothalamus and the anterior pituitary release hormones. Those hormones lead to the release of cortisol, which produces a protective effect on the body. The role of macrophages in immunity is beginning to evolve. We're learning more and more about how these cells have greater and greater roles in protecting us from pathogens. The current known interplay of macrophages in immune responses is illustrated here in Table 18.7. Factors that can tip the scale in the favor of the pathogen are listed in this slide. So the first is malnutrition. And we know that a large number of people around the world are malnourished, which decreases their resistance to local pathogens. The others should make some sense. So pre-existing disease, stress, lack of exercise, not enough sleep, and other types of deficiencies can all lead to increased susceptibility to infection. The last section of the chapter talks about immunodeficiency diseases, and even though these are exciting in themselves, we will not be tested on this material other than to know something about ABO blood groups and rhesus factor. Thank you so much.